The topic of the day is haploid production by means of gynogenesis, ovary and ovule culture. Gynogenesis is a technique of production of haploid plants from unfertilized egg cells. Here, immature ovaries or ovules are excised from unopened flower buds bearing unfertilized egg cells to give rise to haploid plantlets. Based upon whether the explant use is ovary or ovule, it is categorized as ovary culture and ovule culture respectively. Ovary culture. Technique of culturing ovaries in an aseptic condition from pollinated or unpollinated flowers is known as ovary culture. Ovary culture can be utilized for getting both haploid and diploid plantlets. If the ovary consists of the unfertilized egg, it leads to the formation of haploid plantlets, while a ovary with fertilized egg gives rise to a diploid plantlets. The figure on the right shows a vertical section through a carpel. It illustrates the different parts of a carpel. Steps for ovary culture. Healthy flower buds are excised from the mother plant. The developmental stage of the ovary is checked by means of histological techniques. In most of the plants, the optimized stage for ovary culture is a nearly mature embryo sac. The flower buds are then surface sterilized and the outer floral whorls, the sepal, petal and the pistil are removed without damaging the ovary. These are done under sterilized environment. The ovary is then inoculated into the nutrient media for regeneration. The composition and incubation conditions vary from plant to plant and needs to be standardized and optimized. Another method of ovary culture is by means of ovary slice culture. Here, the mature unfertilized flower buds are collected and the developmental stage of the embryo sac is determined by histological techniques. The selected flower buds are then surface sterilized and the ovaries are then cut in transverse section and inoculated into appropriate media. The picture below shows ovary slice culture for propagation of interspecific hybrids of lilium. Steps involved in ovule culture. In this method, the buds or flowers are selected from the healthy mother donor plant. If haploid plants are required, then small unopened buds are excised. For getting diploid plants, open flowers where pollination has occurred are collected. The outer whorls of the flower, like the petals, sepals, and androecium, are removed to expose the ovaries. The ovaries are then surface sterilized. All these steps are done under aseptic conditions inside the laminar hood. After surface sterilization of the ovary, the ovules are then released into the inoculation media. One of the techniques of removing ovules into the inoculation media is known as ovule perforation. In this technique, a small hole is made in each of the ovule without damaging the egg cell or the embryo inside it. It is then inoculated into the cultured media. In many cases, including placental tissue in ovule culture, is found to be beneficial. Ovule perforation method is great help in case of seeds which are large or plants having large seeds. The perforation increases the water and nutrient uptake. The damaged or the underdeveloped ovules are rejected during transfer. After inoculation, the tubes or culture is maintained in optimized conditions for regeneration of plantlets by direct or indirect organogenesis. In ovule culture, two types of support systems are utilized to support the ovules onto the culture media. Firstly, the filter paper support system, wherein the ovules are placed on top of a filter paper placed on the liquid media. Second, the vermiculate support system, wherein the ovules are placed on sterile vermiculate liquid media with the micropilar end of the ovule placed downwards. These methods are successful in case of small seeded plants also. Applications of ovary and ovule culture. It helps in the production of haploid plants where pollen cultures have been unsuccessful. A large number of haploid plants can be achieved at a very short time interval. Pure breeding diploid lines can be created from these haploid lines by mutagenic treatments. This is a great help for plant breeders. In plant breeding, many of the intergenite or distant hybrids give rise to abortive embryos. This could be due to incompatibility issues at the chromosome level or interaction between the endosperm and the developing embryo. Under such conditions, the ovule culture helps in obtaining hybrid seedlings. In some cases, gynogenesis helps in inducing polyembryony. Example in citrus plants. Ovariculture also helps in studying the early developmental stages of embryo and fruit development. It can also be used in studying the effect of phytohormones in parthenocarpic fruit development. Embryo culture. This is a technique in plant tissue culture wherein embryos are excised from the ovary under aseptic conditions and grown in nutrient media to obtain viable plants. Based upon the stage of the embryo, there are two types of embryo culture: mature embryo culture and immature embryo culture, which is also known as embryo rescue method. In mature embryo culture, mature embryos are excised from the ovary and inoculated on the nutrient media for obtaining healthy, viable plants. This is mostly done in cases where the embryos are dormant for long periods. It could be due to certain chemicals in the seed or due to mechanical resistance. In certain cases, the embryos are less survival rate in vivo. In such cases, too, mature embryo culture has helped in getting viable plants. Steps involved in mature embryo culture. In this method, the flowers are excised from the healthy donor plant. Care should be taken that the flowers have fertilized ovules. The flowers are then, under aseptic conditions, are surface sterilized and the ovaries are excised to remove the mature zygotic embryo. These embryos are then inoculated in sterilized, standardized media for growth. Immature embryo culture or embryo rescue. This involves the culture of immature embryos or nutrient media under aseptic conditions to get healthy diploid plants. In many hybridization techniques, the embryos formed are often weak and fail to germinate. This could be due to genetic barriers or due to incompatibility between the endosperm and the developing embryo, which could lead to abortion of the hybrid embryo. Embryo rescue techniques help to rescue these hybrid seeds which fail to germinate. In certain cases, nutritional support for the immature embryos are provided by embryo endosperm transplant techniques. The figure here shows embryo rescue method in chickpea plant. Here, the ovules are dissected and placed onto filter paper bridges in liquid media. Embryo endosperm transplant in case of embryo rescue techniques. Here, first the seeds or the ovules with the hybrid embryo are taken and the embryo is carefully excised out. Another healthy, normal developing ovule with the endosperm and the embryo are taken or selected. In this particular ovule, the normal embryo is dissected out without damaging much of the endosperm. This leaves a normal endosperm with an exit hole. The hybrid embryo is then carefully inserted into the exit hole of the normal endosperm. Embryo endosperm transplant is then cultured in suitable media. The figure on the right shows embryo endosperm transplant technique. Applications of embryo culture. Embryo culture helps in preventing embryo abortion in case of hybrid embryos. In case of many citrus plants, the nucellar tissue gives rise to polyembryos. Under normal environmental conditions, these embryos fail to germinate. By means of embryo culture, mature, healthy plants can be obtained from these embryos. Embryo culture also helps to overcome seed sterility in many plants, like cherries and banana. Embryo culture helps to overcome seed dormancy in many plants like orchids to produce viable seedlings. In certain plants, embryo culture helps in shortening the breeding cycle. The embryos are excised and cultured in vitro to develop into plants in a very short duration. Embryo culture is successful in obtaining haploid plants. Embryo culture is utilized as a viable, reliable, and rapid technique in checking the viability of seeds in seed testing procedures. Embryo culture is also utilized in basic research for studying various growth and developmental stages in an embryo and factors that affect growth. Thank you.